topic, the next uh, adaptation for today, which is atrophy. Okay, so let's look at the definition of atrophy. So definition of atrophy says this is shrinkage of the size of the cell due to loss of cell substance. Okay, easy way to learn atrophy is, you might say, this is exactly the opposite of hypertrophy. What did we learn in hypertrophy? We learned that in hypertrophy, the cell size increases because of production of more proteins. And in atrophy, exactly the opposite happens. Cell size shrinks or reduces because of destruction of proteins. The cell destroys proteins. Okay. So if we agree with that sort of initial concept around atrophy, there are others, right? More mechanisms, but this is the very important principle. How would a cell destroy its proteins? Do you remember of any mechanism of or any cell organelle or any structure inside the cell which can help a cell destroy its proteins? So if you remember something called proteasome and some enzymes called ubiquitin ligases, right? E1, E2, and E3. So what is shown here in this image? Let's look at this image. So this is a peptide chain, and these are chaperones and a folded protein, which is, possibly the active sort of protein, mm, which has been exposed to some aging process or UV radiation or heat or free radicals have damaged this protein. So this has now become denatured or senescent. Okay, so denatured proteins are now acted upon by these three enzymes. It could be E1 is ubiquitin activating enzyme, E2 is ubiquitin conjugating enzyme, E3 is ubiquitin ligase. So all these three, E1, E2, E3, ligases, what they do is add ubiquitin, to this protein chain, and that is like a tagged protein chain which the proteasome identifies, and it gets shredded or broken down into small peptide fragments through this proteasome. So I always like to call proteasome as a protein shredder that is inside the cell. So whenever a cell is undergoing atrophy, protein destruction is basically going on. More catabolism is sort of going on inside the cell. Okay. So this is one of the mechanisms. I'll talk about another one, but this is one of them. So there is an imbalance between protein synthesis and degradation, and an increased catabolism of cell organelles. So the pathway that I just talked about, stimulation of ubiquitin proteasomal pathway, which targets specific proteins and destroys them. So please learn this first. Okay, this is one of the primary mechanisms of atrophy. There is another one, I'll get to it. But let's talk about ubiquitin proteasome. Let's learn this today. Ubiquitin added to a senescent or denatured protein, tags it, it goes through the proteasome. Proteasome will shred it into bits and pieces. Please learn that. That's how a cell is destroying its proteins. The cell which is undergoing atrophy. Okay. Okay. Another thing that you probably noticed in this image here, okay, so this is so this is the endoplasmic reticulum shown to you here in the cell. Now this cell is probably undergoing some metabolic alterations. For example, pH would have changed, or there are some mutations or viral infections have happened. What they do is certain diseases, for example, I mean, we'll talk more about alpha 1, for example, and antitrypsin. That is also one of the good examples. So genetic mutations have happened, and let's assume because of genetic mutations, there is excessive misfolded protein accumulation that has happened inside the ER of the cell, right? This excessive unfolding of protein inside the endoplasmic reticulum of the cell in certain conditions leads to a situation of the cell which is called ER stress. Okay, this is going to activate certain other pathways which are called, sort of unfolded protein response or unfolded protein pathways. We will talk about that later in cell injury. Okay, in the next lecture, okay, or so. What those pathways can do is they can trigger and activate another process, or I would say another pathway of cell death called as apoptosis. So this apoptosis is one of the types of cell death, and this cell is going to die now. In a tissue, if the cells are dying out, the tissue size would shrink. So we will again say that the tissues are, or that organ has undergone atrophy. So now this is somewhat confusing for some of you. Let's clarify. Atrophy, where the cells are shrinking in size, is a reversible change, is an adaptation. But atrophy can also happen, atrophy of the organs and tissues can also happen because of cells dying through this apoptotic pathway. So these are two things that can happen. Because you will now be wondering, apoptosis is not an adaptation. Apoptosis is not a reversible change. Cell death, a cell which died off, is not reversible. So that, of course, is not going to be reversible. Apoptosis, cell death is not reversible, but shrinkage of organ is going to be called as atrophy. So atrophy as such, where the cell is destroying its proteins is a reversible change. But if atrophy has happened because of cells dying, those cells which are dead cannot be brought back. Okay, so that is not reversible. So I hope this is pretty clear. Another thing that we talk when we discuss atrophy is word called autophagy. Autophagy. Autophagy is a Greek word. Auto means self, phagy means eating. A cell eating its own cell organelles, eating its own self. Okay, so why would a cell do that? So autophagy is a process in which a cell eats its own contents. Okay, this might be triggered if a cell lacks certain nutrients. To survive, the cell would have to generate certain nutrients by utilizing its own cell organelles, okay, in an attempt to live on. So that is what autophagy word means, okay. When atrophy happens, when autophagy happens, okay, let's say as an aging process, okay. So aging process is happening, atrophy is happening. A lot of sort of cells are, shrinking and maybe eventually dying off. What will happen is the tissues start becoming more and more brown in color. They start accumulating certain brown colored pigment, okay. And this brown pigment sometimes is inside some vacuoles, inside the cells. And this is another important question that has been asked as an aging process. Okay, so aging process, tissues are shrinking, undergoing atrophy, cells are dying. Why does it become brown? We call it brown atrophy because the tissue starts looking brown, the cells start looking brown. This is because of accumulation of a pigment called lipofuscin within lysosome. So this is also called as wear and tear pigment or aging pigment. Let's make it easier to remember. This word is lipofuscin or fusion, or you can call it, a fusion protein, a fusion sort of lipoprotein. So it's lipids and certain proteins. So lipid fusion with proteins is what this is, right? Let's dive a little deeper into this. So if you look at this image, this is a heart muscle cell here. Okay, so this is showing you certain protein sort of this is showing you this brown colored pigment here. Okay, so atrophy may be accompanied by this sort of brown colored pigment accumulation. So some of the cell debris within autophagic vacuoles sometimes may resist digestion and persist in the cytoplasm within membrane bound residual bodies, okay, and that is lipofuscin sort of granules. Okay, so we'll discuss this later in more detail. Okay, so when this accumulates a lot in these cells, when there is a lot of lipofuscin, so it starts looking brown. Okay, so that is what this word is, brown atrophy. Okay, looks good. So we'll talk more about this in a bit. Again, we did not talk about causes of atrophy. So etiology is one of the pillars that we did not talk about. So physiological atrophy is again common during normal development. So some embryonic structures such as the notochord and thyroglossal duct, they undergo atrophy during fetal development. So those are removed in this process. They undergo atrophy and they sort of shrink and eventually they are sort of removed from the body. The decrease in size of uterus occurring shortly after parturition is another form of physiological atrophy. So initially it underwent hypertrophy during pregnancy. After childbirth, it undergoes atrophy to come back to its normal size, right? So makes sense, I believe. So all the causes from your Robbins textbook have been listed here on this slide. So decreased workload is prolonged immobilization of a limb, complete bed rest, again causing atrophy, loss of innervation. So if nerve stimulation is lost to skeletal muscle, the entire skeletal muscle group where that nerve supplies to undergo atrophy. Diminished blood supply. Diminished blood supply can kill the cells, but diminished blood supply, which is not so much sort of when I say diminished, it's not absent, right? It's not sort of ischemia that is happening. I mean, it's not complete blockage of the blood supply. So little bit reduced can, which is insufficient to kill the cells, can trigger atrophy. Inadequate nutrition, marasmus is another condition that we will learn. This is again classified under protein energy malnutrition. Loss of endocrine stimulation, for example, in hypopituitarism and Sheehan syndrome. So Sheehan syndrome is another important question that has been repeatedly asked in your exams. Okay, we'll talk about this in detail. So hypopituitarism happens, because of infarction, and loss of blood supply that happens. If a lot of excessive blood loss happens during childbirth and or maybe post partum blood loss can trigger a lot of blood supply loss to the pituitary, and that might lead to hypopituitarism and loss of all pituitary hormones. That is what triggers this Sheehan syndrome, right? Aging, we discussed this. So aging affects atrophy in the gonads, vaginal atrophy happens, heart undergoes brown atrophy, thymus undergoes atrophy, brain can undergo atrophy. So aging can lead to atrophy in so many different organs in the body. Pressure, on certain tissues can again lead to atrophy of the cell. So thick secretions, for example, in cystic fibrosis, which can block the ducts, right, can lead to some atrophy in the pancreas. So the pancreas can sometimes shrink because of blockade of pancreatic duct by these thick secretions in cystic fibrosis. Okay. We'll talk about cystic fibrosis as a topic in detail again later. So this picture, again, from your Robbins textbook, shows you cerebral atrophy with, the widening of the sulci here, right, as compared to the normal brain on the right side. So this is, on the left side is the shrunken atrophic brain, and we call this as cerebral atrophy. Okay, good. Let's learn our last adaptation, which is the fourth one, now our metaplasia. So let's look at the definition. So metaplasia is a reversible change in which one differentiated cell type, could either be an epithelial or mesenchymal cell type, is replaced by another cell type. This is the very basic definition of metaplasia. There is more to it, and I will keep talking about this in a bit. Look at this image here. So this is a person who is a smoker, and what I have shown you here is in the top image. This is the normal epithelial lining in the respiratory system. So this is pseudostratified respiratory epithelium, so columnar cells with this cilia on the top, right? So this is pseudostratified respiratory epithelium. In smokers, this converts into what is shown here now is stratified squamous epithelium. So basically what has happened is one mature tissue type has converted into another different type of, in this case, columnar has changed into stratified squamous epithelium. In response, this is an adaptation. In response to smoking, adaptations are reversible. So if this person stops smoking, this epithelium, which has converted into stratified squamous, will go back to its normal, which is pseudostratified respiratory epithelium. Okay, that's what it is. It is reversible. This is temporary change in response to that stimulus. If you take away that stimulus, it's supposed to go back. Okay. If this person does not stop smoking, of course, more bad things can happen. This metaplasia can convert into more like what we call as an irreversible situation called as dysplasia and eventually convert into a neoplasia, that is a cancer. We will talk about that later, okay? But right now I want you to focus on metaplasia first, understand the basics of metaplasia. So again, these are reversible changes in which one adult cell type is replaced by another adult cell type. So how does that happen? How does one cell, when we say one tissue type changes to another tissue type or one cell type changes to another, what's exactly the mechanism of it? This is what is important. So basically what is happening here is reprogramming of the stem cells. Please learn this carefully. It's not a change or an adult mature cell changing itself into another adult mature cell. That cannot happen. What actually happens is the stem cells are reprogrammed. So this is a change at the level of stem cells, and they have been now reprogrammed to grow into a different type of a cell. But the grown-up cell is not converting into a different type. It's the stem cell developing itself into a different type of a mature cell now. I hope this is clear. So metaplasia does not result from a change in the phenotype of an already differentiated cell type. Differentiated cells are the mature cells, right? Rather, it results from either reprogramming of local tissue stem cells or alternatively colonization by differentiated cell populations from adjacent sites. Okay, so please learn this very carefully. A lot of MCQs, they confuse you with what is the very basic mechanism of metaplasia, right? So, and this change is again brought about by various cytokines, various growth factors that these cells produce. Okay. Again, examples are important. So epithelial metaplasia and connective tissue metaplasia are the two subcategories, you can say, or two types of metaplasia. So let's look at these examples. So epithelial metaplasia, a very important example. We keep repeating. So let's talk about this. So this is the most common epithelial metaplasia. Okay, so this is columnar to squamous. Okay, so this is the most common one. Okay, and the example that I just showed you occurs in the respiratory tract in response to chronic irritation. This could be a smoker. So I just talked about that example. So normal ciliated columnar cells, they change into squamous. Okay, so this is the most common scenario when you talk about metaplasia. Okay, so another situation is this vitamin A deficiency. So again, vitamin A deficiency can induce squamous metaplasia in the respiratory epithelium and in the cornea, right? Okay, so it can affect the epithelium in the cornea. It can affect epithelium in the respiratory tract. It can also affect epithelium certain epithelial cells in the kidneys. Okay, so vitamin A deficiency is another topic that we will talk about in nutritional deficiencies, but this is a very good example of epithelial metaplasia. And in this situation now, where columnar epithelium is changing into squamous epithelium, we will call it squamous metaplasia. Okay, if columnar changes to squamous, that is squamous metaplasia. There is an example where squamous changes to columnar, that is columnar metaplasia. So easy way to learn this, easy way to remember this. What it changes into is the final name. So what it finally changes into, right? If the final change after, this switch has happened is squamous, so squamous metaplasia of the final epithelium that is visible in the tissues after this adaptation has happened, that is columnar metaplasia. Okay, if it is squamous to columnar, if it is columnar to squamous, squamous metaplasia, if it is squamous to columnar, columnar metaplasia, both are possible. I'll give you another example. So squamous metaplasia, again, this is the basement membrane, normal columnar epithelium with the cilia on the top in sort of the respiratory epithelium, but now it switches itself into squamous epithelium. So this is squamous metaplasia, as we just talked about. So same thing shown here in this tissue. So this is the columnar epithelium and changing into squamous epithelium now because of smoking or maybe vitamin A deficiency. This is called as squamous metaplasia. Okay, another example. in the urinary bladder. So urinary bladder epithelium is transitional epithelium, okay, on the top, this epithelium here. What you see here in this picture is this is the epithelium on the top, and this sort of urinary bladder wall is infected by Schistosoma hematobium, and that is what is shown here. Numerous schistosoma shown here. So what happens is this infection, this Schistosoma hematobium, what they will do is they are in the lamina propria here. Okay, if you're not familiar with the layers of the urinary bladder, so this is the transitional epithelium on the top. Underneath is the lamina propria, underneath is muscularis and the adventitia. So schistosoma are present in the lamina propria here. What they are doing is they are triggering a change in this epithelium. So the epithelium is trying to survive and adapt to this schistosoma, and it changes from transitional into squamous epithelium. So we are going to call this as squamous metaplasia in this mucosa here. Okay, easy, right? Columnar metaplasia, I told you, is another possibility. So where does it happen? What is the top example that I would want you to remember? Barrett's esophagus. Very repeatedly asked question, high yield topic. So this is a change that we see at the lower end of esophagus in response to reflux of the acid, the HCl, okay, when it becomes excessive and it continuously sort of refluxes back into the esophagus. So at the lower end of esophagus, the cells change from squamous epithelium, the stratified squamous, now changes into columnar to resist and to survive that extra acid that is falling back on them. Make sense? That is, and this change is called as Barrett's esophagus. So it's a switch from stratified squamous in the esophagus, specifically at the lower end, which is now changing into columnar. So this is going to be called as columnar epithelial metaplasia or Barrett's esophagus specifically. And this is an endoscopic sort of imagery that is shown to you here. All this red area that you see on the endoscopy is the change from stratified squamous into columnar. It appears more red. Okay. So that is how the questions will describe it. I'll show you certain questions, but this is what they tell you about in the question. So, okay, so this is about Barrett's esophagus, and this image here is the microscopic biopsy picture. On the left side of this picture, you see stratified squamous epithelium, which is the normal epithelium at the lower end of esophagus, for that matter, like all of esophagus. Right side, what you see is it has changed. It has switched into the columnar sort of epithelium now, right? And you see this side, the right side of this image is all columnar sort of type of epithelium. Left side is the normal one. So there is a switch that has happened at the lower end of esophagus in response to the extra acid that these cells were sort of facing repeatedly, right? Why is this question again important? Because this type of metaplasia, if not reversed in time, might progress to a malignancy, might progress to a neoplasia, might progress into adenocarcinoma at the distal end of esophagus. So there is a risk of malignancy that is increased here because this metaplasia might turn into something called as dysplasia. We will talk about that later. And that dysplasia converts into a neoplasia, which is adenocarcinoma. Okay, right here. Okay. Another example that I can give you here is intestinal type of metaplasia. So intestinal type of metaplasia is seen in the pylorus and antrum regions of the stomach. Okay, and this might have happened because of presence of our favorite bug from microbiology that is called as H. pylori. So in the presence of this Helicobacter pylori or H. pylori, what happens is uh, certain regions in the gastric sort of epithelium, they change into intestinal type of epithelium in response to this bacteria. Okay, so these are characterized histologically by presence of goblet cells. Okay, so they start looking more like the intestinal type of epithelium in the stomach. Okay, so that is the change in response to H. pylori. Another one is connective tissue metaplasia. So this is very interesting. Again, this is formation of cartilage, bone, or adipose tissue in tissues which do not have them. Let's learn this with examples. One of the images that you see here is of myositis ossificans. So myo is muscle, itis is inflammation. So myositis is muscle inflammation because probably of an injury. Ossificans is the word from ossification, which means bone formation. Okay, so myositis is muscle inflammation because of injury, and ossificans means, ossification means bone formation. So this word basically means bone formation in a muscle that has been injured or inflamed. Clear? The name itself told you the entire story actually. So what happens is after sometimes after blunt trauma, so this is how they give you the MCQ and the question. So after blunt trauma, okay, so muscle injury and inflammation has happened. Sometimes during those injuries and as a repair process, certain muscle cells might turn into bone cells. Okay, so I said bone cells. When I say bone cells, this is bony, also called as bony metaplasia. If you want to call it, use that name, please use that. It helps you remember that this is bone formation. This is not just calcium. This is not just calcification. We'll get to that topic tomorrow. Calcification is just calcium deposition in tissues which are injured or dead. Here we are talking about a living tissue which was injured but now has converted into bone. Not only calcium. Bones are not only calcium. Bone is the entire structure, the entire osteoid, not only calcium. So this is different from calcification. Keep that in mind, please. Okay. Do not confuse this with calcification. This is metaplasia. The entire tissue has changed. This is more. Okay. If you want to learn it that way. Okay. Let's learn another example, which is called myeloid metaplasia. So also called as extramedullary hemopoiesis or extramedullary hematopoiesis. Let me explain what this word is. If you're reading this for the very first time, this might seem very intimidating. Like, what is myeloid metaplasia? What is extramedullary hemopoiesis? Let's break this down. Extra in pathology would mean outside. Medullary is the name of the cavity, bone marrow cavity in the bone. So the next word is hemopoiesis, that is production of blood cells. So this word, this name means production of blood cells outside of the bone marrow cavity. Okay. Now, we all know that the bone marrow is the one major tissue responsible for making blood cells. But when we say extramedullary, what are the other tissues that can make blood cells? So this can be seen in liver, spleen, and all long bones. Okay. Liver and spleen do have the capacity to make blood cells. If you've learned about how the blood cells form during, very early on stages, fetal stages, and how it changes from the fetal to the adult form, you might remember that liver and spleen do have capacity to form blood cells, right? So why would this have happened in the adult? Maybe as a disease process, okay, or somebody who is losing a lot of blood cells, and liver and spleen cells, they change into blood forming cells. So that is a form of metaplasia, which has happened. More blood production, blood cell production happens in liver and spleen in those cases. Okay, so that is another example of metaplasia. Let's look at one question before we end our discussion for today. So this is a 60-year-old man with a 40 pack year smoking history, presents with chronic cough. So this is huge, like this is 40 pack, okay. Chest X-ray reveals increased bronchovascular markings, okay. Bronchial biopsy shows replacement of the normal columnar ciliated epithelium with stratified squamous epithelium. We just talked about this. No dysplasia is seen, okay. Of course, we haven't talked about dysplasia, but dysplasia is an irreversible change. We'll talk about that dysplasia word in our neoplasia chapter. Question says, which of the following best explains this observed epithelial change? So I think you would have figured this out. This is a question on metaplasia, but then we need to pick the right answer choice. Okay, so that is where this question becomes difficult. Is it A, clonal expansion of dysplastic stem cells? sorry, with the loss of tumor suppressors? Is it B, differentiation of local stem cells towards a protective phenotype? Is it C, chronic inflammation inducing epithelial necrosis and fibrosis? Is it D, differentiation of terminally differentiated columnar cells into squamous cells? Or is it E, accumulation of DNA damage due to reactive oxygen species? If you want, you can pause the video here and you can try to think of the correct answer. Uh, I'll show you the answer. The correct answer is the B. So I specifically chose this question because sometimes you tend to confuse between B and maybe D. Okay. So the correct answer is B. So this is a reversible adaptation. Okay. I told you in which the stem cells, so that is what changes, right? Stem cells within tissue switch differentiation toward a type which is better suited to withstand the stress. In this case, squamous metaplasia of bronchial epithelium occurs as an adaptive response to chronic smoke exposure. This increases resistance to chemical injury but reduces mucociliary clearance and increasing infection risk. So that is, it comes with, that of course, but of course the cell wants to survive first, right? Can deal with other things later. So option D says transdifferentiation of terminally differentiated. It is not, like I told you, not the terminally differentiated cell which changes to another differentiated cell. It's the change at the level of stem cell. So that's what I wanted you to learn here. Okay. So let's look at a picture from your first aid textbook. Just to quickly recap what we learned today. So these are normal cells, different adaptations that we learned. So hyperplasia, hypertrophy, atrophy. If there is an inability to adapt, what will happen? The cell might get injured. We will talk about that next. If there is a lot of injury, cell might die. So cell death also can happen, right? And we'll discuss more about dysplasia, metaplasia, neoplasia, and later on chapters. Okay, these are not, dysplasia is not an adaptation. Metaplasia is an adaptation, of course it is reversible, but dysplasia and neoplasia are not adaptations. Okay, so please keep this in mind. And again, I have one slide for you. Please read this and remember this always. So these are what happens in metaplasia. So this is resident tissue stem cells or basal progenitor cells which are reprogrammed or which change. So these stem cells differentiation, down an alternative lineage. In this case, in our question, it was, squamous instead of a ciliated columnar, right? So the mature cells do not transform. That is a wrong option. That was the wrong one, right? I hope this is all making sense and this is all good for today. What I wanted to quickly also share with you is our Robin's textbook pages that I have got this information from, so that you can also quickly look at your Robbins textbook. And I did promise you that we will read our Robbins. So this is page number 52 in your 11th edition of Pathological Basis of Disease. Okay, uh, so this is the topic on adaptations of cellular growth and differentiation. So we talked about, these are changes in the size number. Then we talked about hypertrophy, the definition of hypertrophy. Then we looked at, pathological hypertrophy and its examples, right? Muscles undergoing increased workload. Then we talked about this one, heart in response to hypertension, right? Then we looked at physiological hypertrophy and smooth muscle fibers and uterine hypertrophy during pregnancy. We talked about this. So mechanisms again is increased protein production. We talked about that. We looked at these images. We looked at this image, right? So we have covered all of this and the description of what are the certain genes which are activated, what are certain growth factors, and what are some transcription factors. If you want, you can learn these names, right? So cardiac hypertrophy genes, we talked about that, the beta isoform, alpha isoform, right, switch that happens. Then we also talked about hyperplasia, right? So hyperplasia is increasing the number of cells. So physiological hyperplasia, we talked about this, examples, uh, female breast at puberty, right? Then we talked about pregnancy. We also talked about this example on partial hepatectomy where it is called compensatory hyperplasia. Then we went to talk about, the bone marrow. Then we talked about pathological hyperplasia, which is the example that I gave you, right? So abnormal hormone-induced hyperplasia. So the endometrial glands undergoing hyperplasia, right? So we covered all of this. Then next is, sorry, hyperplasia. I told you there is increased risk of cancer. So we talked about that. Viral infections, we looked at all of this information. Mechanisms, again, growth factor driven. So we covered all of this. Atrophy, I told you, all the different causes. So they have listed the causes. So decreased workload, loss of nerve, okay, diminished blood supply, in nutrition, loss of endocrine stimulation, pressure. So we looked at all these examples. We saw this image, normal brain versus the atrophy in an 82-year-old, right? So we saw that here. We also talked about the ER stress and sometimes the cells undergoing apoptosis, and that also causes tissue shrinkage and organ shrinkage. So mechanisms, we talked about the ubiquitin proteasomal pathway, we talked about autophagy, residual bodies, lipofuscin, brown atrophy, right? We talked about those concepts. Then we jump to metaplasia. So what is the definition? What is the most common type? Right? We talked about all these examples, metaplasia from squamous to columnar. We talked about connective tissue. We talked about myositis ossificans, right? We covered so much today, right? So metaplasia definitions, and we clarified this concept through an MCQ. Okay, we talked about vitamin A deficiency, what retinoic acid deficiency does. So we covered 4 pages from Robbins in almost a little over 1 hour today. So all of this information in Robbins is the sort of key concepts that we learned. So you can review this. So this is page numbers 52 to 56 that were covered today, right? All of them are covered today. So you can very confidently now open your Robbins textbooks and please look at all of this material. Okay, and I will also post some reading material you can use to sort of learn these topics in depth, and you can find that in the description. You can download that file. Okay. Anything else, if you need, please feel free to reach out on my email and feel free to comment on this video, and I will try to respond as soon as possible. So thank you everyone. I will see you in the next video and take care.